So far we've been talking about cells. What is inside the cells, like the organelles and what they do, what cells are made of, the proteins, nucleic acids, carbs, lipids, and most recently we talked about the cell cycle, how we make more cells. Well, today we're going to focus on just the nucleic acid side of it, just the DNA. So the first thing we have to do is look at our structure of DNA. We already know that DNA is a biomolecule and its monomer is a nucleotide. So we're going to box where we see the nucleotide. We are going to draw a box right around these three components and label it nucleotide. When we look at a nucleotide, we see that there are three different parts to that nucleotide. So there's this little circle, which is called the phosphate. We're going to put a P. There's the pentagon, which is the sugar. We're going to put an S. And there's this thing connected in the middle. That is the nitrogenous base. I'm going to put N base. So those are the three parts of a nucleotide. Now, we know that DNA has four letters, A, T, G, and C. A always pairs with T, and those are going to have two bonds. So when we look at our picture, our top picture has two bonds. So that has to be our adenine and thymine that go together. Down here we see three bonds. That has to be our guanine and cytosine. Again, adenine and thymine each have two bonds. Guanine, cytosine each have three. That's what DNA looks like. When we look at just this side, we call that the sugar phosphate backbone. This is basically what makes up the sides of the DNA. Everything else in the middle are those stairs. Those A's pairs with T's and G's pairs with C's. So the DNA structure we just looked at is really important because it helps it perform its function. Remember, DNA, the functions are to store genetic information and to code for proteins. So now we're going to talk about how these DNA molecules are going to code for proteins. So always, we're going to start with that double helix of DNA. So let's go ahead and label our DNA double helix. If we want to make more DNA, then all we're going to have to do is DNA replication. When we do DNA replication, we're still going to keep those same four letters: adenine, thymine, guanine, and cytosine. A pairs with T, G pairs with C, and vice versa. Now, when we want to start protein synthesis, we are going to make a strand of mRNA, and that's going to be this strand right here in the middle. RNA is single-stranded. RNA is different because it still has adenine, guanine, cytosine, but instead of thymine, we see a U for uracil. So in this case, in RNA, when we want to make that, A is going to pair with U and G with C. So let's go ahead and label this middle molecule. That's the mRNA. Now, when we go from DNA to mRNA, the process we call that is going to be transcription. Transcription takes DNA and makes mRNA. Then, if we want to take that mRNA molecule and make a globular protein, then what we're going to do is translation. So we're going to make our protein from our mRNA, and we are going to call this translation. When we want to make a protein, when we're actually trying to do this on paper, that is where our codon chart comes into place. So write the word codon chart. That's going to remind us that is what we use to find things like methionine or leucine or all of those other amino acids we have. Now let's take a look at how we kind of see this protein synthesis happen inside of a eukaryotic cell. So here's our eukaryotic cell. We know it's eukaryote because we see this nucleus holding our DNA. So again, we're going to start with DNA. When we move from DNA to mRNA, that's going to be transcription. So go ahead and write down transcription. Transcription, because the DNA can't leave the nucleus, it has to happen inside the nucleus. Transcription happens in the nucleus. Now, if I'm a prokaryote, obviously it has to be in the cytoplasm because I don't have a nucleus. Now, the mRNA is a special little molecule. It can actually leave the nucleus. It's not as delicate as DNA. So when we send that mRNA out, we are going to send it over here to a ribosome. And the ribosome is where we are going to take that mRNA and translate it into a protein. And the name of that process, as we remember, is translation. And translation is the one that happens out here in the cytoplasm. So again, we start with DNA, make mRNA, that's transcription. Happens in the nucleus. Take the mRNA, send it out to a ribosome for translation, and that happens in the cytoplasm. And remember, if I'm a prokaryote, no nucleus, everything happens in the cytoplasm.